Hi, Assalamualaikum warahmatullahi wabarakatuh. We continue on our chapter 12, plant hormones. Okay, at the end of this topic, you must be able to understand different types of plant hormones and their functions. So for introduction, hormones, they control plant growth and development by affecting the division, the elongation of plants, and also the differentiation of cells in plant. So these plant hormones are produced in very low concentration, and it is enough for the plant to grow and to develop an organ. So the first hormone is auxin. Okay, so this auxin is function to promote cell elongation in different target tissues. So according to a model called the acid growth hypothesis, okay, it involved the proton pump activity. So this is the plasma membrane and this is the cell wall. So when there is an auxin, okay, when there is a presence of auxin, what happens? Auxin will cause or will stimulate the pumping of the hydrogen ion from the cytoplasm to the cell wall. Okay. So what happens when the ion of hydrogen pump to the outside? Okay, so when there is more hydrogen outside or in the cell wall become more acidic. Okay, so this acidity will loosen the cellular structure. If we already know the cellulose is a very rigid and tough structure, when there is acidity increase in the cellulose area, the structure of the cellulose will be loosened so that the cell can be elongated. So this is the acid growth hypothesis. Okay, so you can read this. This is in details. So the auxin increase the activity of proton pump from the cytoplasm to the cell wall, and then the cell wall become more acidic. What happened then? The wedge-shaped expansin. Expansin is a type of protein, but it is non-enzymatic protein. So this expansin only activated when there is a low pH. Okay, when there is low pH, meaning more acidic, the expansin will be activated. This expansin will separate the cellulose microfibril. Okay, so this is the microfibril. Okay, it will separate the cellulose microfibril from the cross-linking polysaccharides. Okay, the microfibril is linked by polysaccharide. So this expansion will help to lose the cross-linking polysaccharide, and the exposed cross-linking polysaccharide are now more accessible to the cell wall enzymes. Okay, so now cell wall enzyme take over. Okay, so this enzymatic cleaving of the cross-linking, okay, so this is the cell wall enzyme, allow the microfibril to slide. Okay, to slide meaning to move. The extensibility of the cell wall is increased when there is a turgor. Okay, when there is a the movement of water inside of the plant cell, so it will increase the turgor pressure, and then the cell can be expand. With the condition of the cellulose loosen, the cell can be elongated. So this is roughly the description of the acid growth development. Okay, so this is another role of auxin in lateral and adventitious root. So it's involved in the formation and branching of roots. So auxin promotes elongation. Normally it occurs in the part that need elongation, such as the shoot and also the root. Okay, another function of auxin, it can also function as herbicide. Okay, an overdose use of auxin can kill the eudicots. And auxin in secondary growth, if you still remember the secondary growth, which involve the vascular cambium. Okay, so this vascular cambium will be differentiated and it forms the secondary vascula, which are the secondary xylems and secondary phloem. So this is the vascular cambium. Okay, so it will differentiate and then they form secondary xylem here and secondary phloem. So auxin also promotes secondary growth. Now we go to the second hormone, cytokinins. Okay, so the function of cytokinins to stimulate cell division, and they are produced actively in growing tissues such as the roots, the embryos and the fruits. And these cytokinins work together with the auxin. So this is the chemical structure of the cytokinins. Okay, now we go into the control of apical dominance by cytokinins. Cytokinin, auxin and other factors interact in the control of apical dominance. Apical refer to the apex. Okay, apex mean at the end, either at the shoots, either at the roots. Okay, the ability of a terminal bud to suppress development of axillary buds. So this is another functions of cytokinins. If the terminal bud, okay, so just if the terminal bud is removed, the plant become bushier. So this is one step. Okay, normally gardener they cut the terminal bud in order to make the plant more bushier. Okay, and cytokinin also have anti-aging effect. Cytokinin retard mean stop. Okay, retard the aging of some plant organs. How? By inhibiting the protein breakdown, stimulate the RNA and protein synthesis, mobilizing nutrients from surrounding tissues. Okay, so this is the action of how cytokinin retard the aging, stop the aging of some plant organs. And the florist, florist will use this cytokinin spray to cut the flower fresh. So this is you may use this as the application of cytokinins in industry. So we go into third hormone, gibberellins. Okay, so gibberellin have a variety of effects such as stem elongation, the fruit growth and germination. For the stem elongation, gibberellins stimulate both of the leaf and also the stem. Okay, in stem, gibberellins stimulate cell elongation and cell division. So just don't be confused. Okay, within the auxin and also gibberellin. Gibberellin refer to the more focus to the stem elongation, but for auxin they more to shoot and root elongation. Okay, now gibberellins in fruit growth. Okay, in many plants, both auxin and gibberellin must be present for fruit to set. So in commercial, this gibberellin hormone is used in the spraying of Thompson seedless grape. Okay, how? Okay, so please search on this. Okay, how these gibberellins are used commercially in the spraying of Thompson seedless grape. Okay, so this is the process of germination, seed germination by gibberellins. After water is imbibed, okay, there is release of gibberellins from the embryo. This gives the signals of seed to break their dormancy and germinate. That's why when we plant something, okay, when we plant seed, the seed needs more water. So that's why we need to plant the seed with numerous amount of water in order to break their seed dormancy and cause or to start the germination process. Okay, so this is the process. After a seed imbibes water, the embryo release gibberellin as a signal to the aileron, the thin outer layer of endosperm. Okay, so that is the functions of water to make the embryo release the gibberellin. Okay, the aileron respond by synthesizing and secreting digestive enzyme that hydrolyze stored nutrients in the endosperm. One example is amylase, which hydrolyze starch, which is a similar enzyme in our saliva, helps in digesting bread and other starchy foods. and the sugars and other nutrients absorbed from the endosperm. Okay, remember the endosperm is the tissue that supply nutrients to the plant. Okay, sugars and other nutrients absorbed from the endosperm by the scutellum. Scutellum is the cotyledon, the seed itself, or consumed during growth of the embryo into a seedling. So this is the process of germination. How germination is start by the presence of gibberellins, and they get their food from the seed itself, okay, from the endosperm tissue. Okay, the fourth hormone is abscisic acid, okay, for seed dormancy. Two of the many effects of abscisic acid are the seed dormancy has great survival value. So what is the function of seed dormancy? To make the plant survive. In order the condition is non-favorable. Okay, so this seed dormancy will ensure that the seed will germinate only when there are optimum conditions. The high level of abscisic acid, ABA, is the short form of abscisic acid, in maturing seed inhibit germination. When there is abscisic acid, the seed will be not germinated. Okay, so the function of abscisic acid is contra to the previous hormone, which is the gibberellins. So this is the structure of the abscisic acid. Okay, so this is another condition of abscisic acid, drought tolerance. Okay, abscisic acid is the primary internal signal that enables the plant to withstand the drought. When plants begin to wilt means not fresh. Okay, when plants begin to wilt, the ABA or abscisic acid will accumulate in the leaf and cause the stomata to close rapidly, so that no more water will be lost. Okay, so this is to prevent further water loss, and the process of transpiration will be reduced. Okay, so this is another function of abscisic acid during drought season. Okay, the fifth hormone is ethylene. So plant produce ethylene in response to stress. Okay, what kind of stress? Such as drought, flooding, mechanical pressure, injury like cut, and infection. So this is the stress that normally happen in plants. Okay, when plant facing stress, they will produce ethylene hormone. Okay, ethylene induces the triple response, which allows a growing shoot to avoid obstacle. In the triple response, what happened? The stem elongation will be slow. The stem become thickened so that it become stronger. The curvature causes the stem to start growing horizontally. Okay, please search on curvature. What is curvature? Okay, search on this. Okay, so this is one experiment that have been conducted to test the triple response towards mechanical stress. Okay, the experiment, germinating pea seedling were placed in the dark and exposed to varying ethylene concentration. So they used variation in the concentration of ethylene. Their growth was compared with a control seedling and not treated with ethylene. Okay, so the control plant is not treated with ethylene, and the rest is treated with different concentration of ethylene. So let's see the results. All the treated seedlings, okay, so this is the concentration. This is no ethylene. This is 0.1, 0.2, 0.4, and 0.8. All the treated seedlings exhibited the triple response. Response was greater with increased concentration. Okay, when there is increased concentration, so the response towards the growth also increase. Okay, so it stopped the growth of the plant. So ethylene induced the triple response in pea seedling with increased ethylene concentration causing increased response. That's why when there is more ethylene, the response also increase. Okay, the response of plant. The response refer to the growth of the plant. Okay, so this is about ethylene. insensitive mutants fail to undergo the triple response after exposure to ethylene. Constitutive triple response mutant undergo the triple response even in the absence of ethylene. Okay, so this is just some experiment that have been done to test the functions of the ethylene. Okay, apoptosis. Apoptosis also refers to the cell death. A birth of ethylene is associated with the program destruction, okay, meaning the cell's destruction. Okay, this is a test on the cells and organs and also the whole plants. Okay, a change in the balance of auxin and ethylene control leaf abscission. Okay, the process that occurs in autumn when leaf falls. So the leaf falls also related to the functions of auxin and also ethylene. Okay, this is another function of ethylene hormone. And also, ethylene also induce fruit ripening. Okay, a burst of ethylene production in the fruit triggers the ripening of fruits. So what happened in the process of fruit ripening? The enzymatic breakdown of cell wall component soften the fruits. Conversion of starch and acid to sugar makes the fruit sweet. Normally in industry, when the plant, okay, the botanist or the industrial, okay, they want to ripe a fruit faster, they just use ethylene hormone, okay, in order to fasten the process. So this an overview of the plant hormone that we have been discussed. There are 6 hormones here. The auxin, also short written as IAA, the cytokinin, the gibberellins, okay, abscisic and ethylene. Okay, so there are 5 only. Okay, sorry, there are 5 hormones, and this is the place where they are produced or found in the plant. Okay, and their major functions. Please note on these 3 points: the hormone, where they belong or they found in the plant, and